Thank you. Hello, everyone. Thank you to the organizers for this opportunity. I will show you now a slightly different perspective, which is how we envision the industrial challenges for moving mRNA vaccines from the research stage to the commercialization stage. If I can have the, oh, that's me. That's me again. So I always like to start with a little bit of a historical perspective and with a few landmark papers on the gene transfer and the non-viral vectors that have been used to transfer gene material into animals. Okay, this was one missing. In particular, this one, which was published now 30 years ago on the bottom right of the slide, which was, to my knowledge, one of the first demonstrations that injecting mRNA in mice encapsulated with liposomes, you can induce an immune response in animals. And this was published by Pierre and his colleagues in a company called Pasteur-Mérieux Serum et Vaccin, which is one of the predecessor companies to the company I am representing today, which means that the roots of these mRNA vaccines have been embedded in this country for quite a while now. And this is good to see the progress. So the mRNA Center of Excellence has been created in 2021 by the addition of, as I said, the expertise of Sanofi Vaccines in the industrial development of vaccines, and the expertise of Translate Bio, a biotech company in Massachusetts, specialized in the design and the delivery of mRNA. We are located in two areas, mainly in the US and in France, about half in the US and half in France, and we are taking advantage of the ecosystem and the innovation clusters that are existing around Cambridge on the one hand and around Lyon on the other hand. We are now more than 500 people devoted to the development of mRNA vaccines, incorporating all the necessary stages from the early research to development, clinical development, and industrialization of mRNA vaccines. Since our creation in 21, we have put around 10 mRNA LNPs in clinical trials, and we are preparing to put more in clinical trials this year. And I will going to show you already the first examples that the platform that we have introduced is already working quite well in clinics. So this is one example on the left with the influenza virus, where you see the response to the injection of mRNA LNPs with a good response to the A strain. You see that the base strain, the B strain, which is where is this? Which is here, sorry. We see a suboptimal response to B strain, which has been already published by other companies. And this is, as you see, also the case with the standard of care, which is showing a lower response to the B strain. With another respiratory virus, which is the RSV virus, we see also a very nice response in different populations. So this is for the immune response. An important aspect is the reactogenicity of the mRNA vaccines. And as you can see, we have higher reactogenicity than the standard of care and higher reactogenicity than, of course, with the saline. But we have a quite reasonable type of reactogenicity with only moderate and limited number of grade 3 reactogenicity events in humans. So this is very encouraging data with our very first platform of mRNA vaccines. And I'm going to show you what are the challenges which are still ahead of us now. There are three main challenges with the current LNP technologies. The first one is the potency, which as shown you needs to be increased for certain antigens. The second one is the reactogenicity or tolerogenicity that I just showed. And finally, the thermostability that we'll not have time to describe today, which is very important if you want to have vectors that can be stored in reasonable conditions for daily use, especially in large indications like flu or other respiratory diseases. And I'm going to show you the tools that we are using in two areas, delivery and translational models using machine learning tools to address the challenges of potency and tolerability. You know, the LNPs are based today on 4 components: the cationic lipid, the helper lipid, a sterol, and a pegylated lipid. And we are not only developing new cationic lipids, but also developing new helper lipids that, as you see here, can trigger an improved response after injection in animals. We are also developing new steroids that also give a significant enhancement of the potency in animals. And finally, we want to add novel excipients that trigger, again, an improved response. And by combining all these elements into a classical chemical design and chemical optimization approach, we hope that we can improve the efficiency of the delivery of mRNA in animals and then in humans. The second part that we need to work on is the reactogenicity, as I explained before, which is still higher than the standard of care. We are starting from an in vitro system, the mimic system, proprietary system, which is a kind of immune system in a tube. We are treating PBMCs with the LNPs, and we measure the cytokine response to these LNPs on the one hand. On the other hand, we are exploiting all the clinical data that we get using all the same LNPs and the signatures that we have in humans in response, and which are signatures of reactogenic events in humans. And by plugging a machine learning system in between, we've been able to correlate the two elements, the in vitro to the in vivo human reactogenicity, and to build a model that allows us to predict the probability that a given LNP will elicit a reactogenic event in humans. And this way we can rank our LNP candidates or our formulation candidates, and of course select the one that has the best chance of showing a low reactogenicity in humans. So these two examples show you the current challenges that we are addressing in terms of efficacy on the one hand and reactogenicity on the other hand. And we have also to prepare for the future. So what is the future for us from an industrial standpoint? The future, as I said, since we have a good platform which is already working in clinics, but which is giving us some suboptimal answer to some antigens, we need a second generation platform which will be more potent, based, for example, on the development of new lipids, as I showed you. We need to continue to use the machine learning models to improve the performance and to decrease the reactogenicity of the mRNA vaccine that we are introducing into patients. Once we have solved this problem, we will be able to extend and expand the use of mRNA viral vaccine that I showed you today, with the examples of flu and RSV, to other antigens like bacterial antigens, and we are preparing for clinical trials in the acne area or chlamydia infection area. And we are also starting research programs outside of the vaccine business unit with the possibility of using mRNA beyond vaccines for therapeutic applications. We don't do this in isolation. We are constantly opening to advices from external experts through regular advisory boards. We have quite a few number of collaborations with academic institutions, some of them being located in this very city. And we also are sponsoring a number of awards that you may be aware of. As mentioned by Dr. Pichon before, the manufacturing of mRNA LNPs involves different steps, different intermediates. It's a quite complex process with, at the end, mRNA drug substance, a lipid nanoparticle, and a final drug product. And all these steps have to be, of course, well anticipated and scale up to prepare for the first commercial launch and for the large-scale use. You can imagine in the field of flu, for example, that a number of products to be manufactured will be quite important. So this tech transfer has to be anticipated years in advance, and we are already anticipating the technological transfer of these technologies from the laboratory to the commercial facilities. And for example, the technical transfer of the mRNA manufacturing to our facility in Lyon area has been already started. This is a completely new facility, which is a modular state-of-the-art and fully digitalized facility that will be used for the manufacturing of future mRNA drug substance in this country, and this we are already preparing for. Similarly, we are preparing for the tech transfer, and we are initiating the tech transfer of the LNP and the final product manufacturing, be it vials, prefilled syringes, liquid or lyo, in our facility in Normandy. And I will stop here, and looping back to the very first slide where I showed you the paper published 30 years ago for the same company. It's good to see that the roots that we have planted 30 years ago are started to deliver some fruits. And this has been a very interesting adventure, technological, scientific, and human adventure to develop these mRNA vaccines. And I thank you for your attention.
Thank you, Pierre.